Hello everybody, thanks for listening and thanks to SCAR for giving us the opportunity to present our talk. I'm Annick Belgium, Director of the BCCM ULC culture collection of Cyanobacteria, and here are our colleagues. The talk will be divided into two big parts, one about the collection history and objectives, and then its diversity, genomics, and exploration of the bioactivity potential. So let's start with the first point. Since 2006, the culture collection has evolved from an in-house research collection into a public one that is research-based, providing services open for legitimate users, operating within a legal framework, and linked to the Belgian Coordinated Cultures of Microorganisms, BCCM. So what is the BCCM consortium? It's 7 decentralized culture collections together coordinated by a central team at the Belgian Science Policy Office in Brussels. So here you see the central cell and bacterial, fungi, mycobacteria, diatoms, plasmids, and here we are with the cyanobacteria. Culture collections are important because we publish and then we move labs, we change jobs, or we perish. And therefore important biological material and related information might be lost forever. And it's not a joke. Here is an extract of an email I received. Hi Annick, unfortunately I don't have them, strains, either in the move to somewhere about 5 years ago. I think they did not move over. Sorry about that. So our collection has actually 246 unicyanobacterial strains, of which 120 of pole origin. They are maintained as living cultures with a cryopreserved backup. We have an ISO 9001 certificate for deposit and distribution services in the frame of a general certificate for all BCCM collections. We have also the possibility to have safe deposits, which means they will not be in the public catalog, but it's a kind of backup for laboratories who are afraid to lose important strains. There is a geographic focus, of course, on cyanobacteria from polar regions, and we try to have a large diversity representative of the whole cyanobacterial realm, like Synechococcales, Coccidiopsiales, Nostochales, Oscillatoriales, Plurocapsales, and Synechococcales. The quality control of purity and authenticity is made first by microscopic observation and then by molecular markers like 16S and ITS sequence. Concerning the second part, we have built a tree based on 16S ribosomal RNA sequences for the culture collection on 206 strains, and this means that we could observe 82 OTUs at 99% similarity threshold, of which 45 were of polar origin. So now we are going to look at the diversity cluster by cluster in more details. I have to tell you that the taxonomy of cyanobacteria is still in revision, and I would say in a quite confused state. That's why in some clusters you will find different names. So here I present a cluster that I would call now Microcoleus, but in the past names like Formidium autumnale have been used. Here you see 20 Antarctic strains and 5 Canadian ones, of which 3 from the Arctic region. Another related cluster includes 9 Antarctic Formidium autumnale strains from McMurdo Ice Shelf and James Ross Island. And 2 Brazilians are related but somewhat distant. Another cluster is made of Filamentous genera, 10 from the Antarctic and 5 Canadian strains, of which 2 Arctic in this cluster. Another cluster of Filamentous genera is gathering 12 Antarctic strains. You see, for example, a big group of Shackletonellia antarctica, Timaviella, Pegetrix. Here, another group with 3 lineages from polar regions, with 18 Antarctic strains and 1 Arctic one. There is a group of Stenomythos, Plectolingbia odsoni, that we are going to discuss in more details, and Formidesmis priestley, which has also some interesting features. So Plectolingbia odsoni. In 2011, we participated to the description of a new genus and new species. And in fact, just now it looks like it's a potentially endemic genus, as it was till now only found in Antarctica, in Pritz Bay and McMurdo Dry Valleys. One strain of that group, ULC 009, has been tested against Candida kreutzii and showed a growth inhibition of the fungi by the secondary metabolites that were in the purified extract. This was data obtained in bilateral cooperation with Brazilian colleagues. Now let's go back to this part of the tree, but we are going to look in more detail into the Formidesmis priestley, where you see 007 and very closely related, because in fact it comes from the same lake in the Larsemann Hills, 026. The draft genome of ULC 007 was published a few years ago and is the first genome sequence of an Antarctic cyanobacterial strain. Its total length seems to be close to 5,700,000 base pair, and annotation indicates that there would be 5,600 protein encoding genes. If you look at the repartition of the gene copies into the subsystems of RAST, RAST is rapid annotation using subsystems technology, you see, for example, that there are a lot of gene copies related to carbohydrate, for example. There was a comparison of the sequence of ULC 007 with another Formidesmis priestley from the Arctic, Loptolingbia boreana, and 69 complete and representative genomes of cyanobacteria from other taxonomies and other origins. Here you see graphs where you have taxonomy, so the family Synechococcales on the x-axis, and the number of copies of genes belonging to 4 different subsystems which are supposed to be involved in cold resistance. And indeed, in blue are the 2 polar strains, and you see that even in their own family, Leptolimbiaceae, they constitute the most abundant gene copies. So in polar regions, we know that low temperature induce disturbances like rigidity of membranes, reduction of the rate of enzyme catalyzed reaction, inefficiency of solute transports. Therefore, these harsh conditions select organisms that present particular adaptation. A result underlines the presence of functional categories of genes involved in the production of key molecules like EPS, fatty acid, and chaperones. These molecules limit the cellular and biochemical damages like EPS, protecting against freezing by buffering temperature fluctuation around 0 degrees. Some fatty acids decrease the membrane's rigidity. And so the success of ULC007 could be explained by different functional processes, such as exopolysaccharides, biosynthesis, DNA repair, osmoprotection. So ULC026 is very related to 007, and its bioactivity against 3 fungal strains were assessed. And in 2 cases, you could see an antibiotic effect. Another cluster groups 9 Antarctic strains of Filamentous genera. Another group of Filamentous genera includes 11 Antarctic strains. So they are also mingled with Belgian and Italian ones, but they come from different places in Antarctica. Then in the Synechococcales order, we have 10 Antarctic strains and 1 Arctic one. So here you see them, and there are a lot of Cyanobium of different origins in this group, but the polar ones are somewhat separated from the temperate. And the last group includes the Coccidiopsis and Nostocales, and there are 28 strains in that big group, with also Asalia and a lot of nostocs from 2 regions in Antarctica. And one of them, ULC050, has been tested for its bioactivities. During the European project Micromat, some strains were already tested for bioactivities, and nostoc ULC050 that we just saw was active against Staphylococcus aureus. Interestingly, the strains ULC026 and ULC009 had already been detected as having bioactivities against different bacteria and fungi. So there is a renewed interest for biotechnological exploitation of cyanobacteria, with a growing number of strains found to produce novel and biologically active compounds. Current discussion at the ETCM level concerns the exploitation of Antarctic genetic resources and how this fits with the Antarctic Treaty, with the Madrid Protocol. And Belgium has been active on these topics from 2007 to 2014, with a number of working papers and information papers. And even in 2009, Belgium and UNEP have decided to create a database on bioprospecting activities, which was called Bioprospector. It was developed by the United Nations University Institute of Advanced Studies, based on public information, mostly patent, and it was freely accessible on the internet. However, it was discontinued due to lack of financing. But in 2009, there were 187 records already. And the purpose of the database was to show that really Antarctic genetic material was really used in industry and in different type of productions. And here you find the different uses of the genetic resources. There was also an IP on the role of ex situ collections, so culture collections in Antarctic bioprospecting. So many thanks to the numerous colleagues who have deposited their precious strains into the collection, and to the team of Marli Fiore in Brazil that has carried out the most recent bioassays. And thanks to you for your attention.